Welcome to our podcast on the male reproductive system. As we move through our male reproductive tract, we're going to notice that we begin with spermatozoa development in the testes, and we're going to move into the abdominal cavity through our genital ducts, and then we're going to exit the body through the urethra, the common pathway for sperm and for urine. And so much of what we're going to do today is going to involve the testes and identifying the different regions of the male testes, as well as identifying the different stages of spermatozoa development from spermatogonia through spermatids to spermatozoa. Then we're going to identify the layers of the male reproductive tract. Now, this is going to be critical that we're able to compare and contrast the regions histologically to distinguish efferent ductules from vas deferens, from prostatic urethra, and all these different regions throughout the genital tract. As we begin to get to the lower reproductive tract, we're going to see the urethra pass through the prostate gland, and so we're going to look histologically and be able to distinguish the prostate gland from other glandular organs we have in the body. And then lastly, we'll look at the erectile structure as well as the urethra that's present in the male penis. So once again, we're going to begin with the testes. We'll look at the genital ducts, both intra and extra testicular ducts. We'll look at the associated glands, principally the prostate gland, and we'll end our discussion with the histology of the penis. Here we can see in a larger diagram the overall view of male reproductive tract. Again, we begin in the testes. We have the epididymis, this coiled cord that's adjacent to the testes before we move into the vas deferens, which re-enters the body. This then is going to go into the seminal vesicle. Now, the seminal vesicle is going to be attached to the prostatic urethra through this short ejaculatory duct. They've made it quite a bit longer for illustrative purposes. And here we can see the prostate gland surrounding the prostatic urethra just as it leaves the bladder. We're also going to have a bubble urethral gland that's going to contribute some components. And then we get into the urethra proper as we get into the penile urethra, and that then will allow our genital material to exit the body. Now, as we begin with the testes, on the outer portion of the testes, we're going to have our peritoneal layer, and this is going to be the tunica vaginalis. Immediately beneath is the tunica albuginea, and we see this same structure in the female ovary, and in fact, the female ovary and the male testes are going to develop from the exact same tissue. Early in your developmental processes, there was an indistinguishable period. If you looked at ovary versus testes, you couldn't tell one from the other. So it's a very similar architecture here, even though the end products and their function are considerably different. Now, just beneath the tunica albuginea is going to be a highly vascularized layer of loose connective tissue, and that's the tunica vasculosa. But the tunica albuginea is going to invaginate within the testes itself, and this is going to divide the testes up into different lobules. Now, the very middle portion of this septa is going to be the mediastinum testes, and we'll see that in a couple of different slides. But we'll have smaller septa that divide the testes into a number of smaller lobules, and each lobule then is going to contain the first part of our genital tract, and these are going to be between 1 and 4 seminiferous tubules. Here we can see an illustration of the male testes, and you can see how the septa have divided this into smaller compartments, and with each one of these lobules, we're going to have a number of seminiferous tubules. In a histological cross-section, we can very easily identify the tunica albuginea, this thick, fairly dense connective tissue on the outside. The vaginalis is going to be external, and the vasculosa will be just deep. But here we can see some of the connective tissue septa diving in. This is the mediastinum testes, and here are the smaller septa that are dividing this up into different compartments. Within these compartments, then, these lobules, we'll see cross-sections of our intratesticular ducts, the beginnings, the seminiferous tubules, and here you can see many of those. Within these sections, we can see a number of nuclei, the majority of which are going to be spermatozoa at different stages of development. We will also have some supportive cells, generally located toward the base, and these are going to be our Sertoli cells. So in this illustration, we have some spermatogonia at the very base. These are our mitotically active germ cells, regenerating cells. Here we have these large Sertoli cells. These are nurse supportive cells, and we'll define them and their function a little bit later. And then here we can have some spermatocytes and spermatids, different stages of development. The youngest cells are near the base, whereas the more mature cells are ready to move out into the extratesticular ducts. They'll be closer to the lumen of our seminiferous tubule. So here is our path of sperm. This is going to represent our genital tract. The seminiferous tubules, tubuli recti, and the redi testes, these are going to be intratesticular, the first three. Efferent ductules are going to interconnect the intratesticular ducts with the extratesticular ducts. So the efferent ductules are almost like an intermediate between the two. And then the epididymis, vas deferens, and the urethra are our extratesticular genital ducts. Now, just a little bit about vascular supply. Certainly we're going to have a testicular artery and vein, but this is a quite interesting arrangement. The scrotum serves the function of temperature control. Sperm must be kept within a fairly narrow range of body temperatures. Too hot, too cold, and you lose viability, you lose motility. And so typically in a fertility clinic, if a couple is having difficulty conceiving a child, one of the first things they're going to suggest to the male is to wear boxers to allow the scrotum to more easily regulate the body temperature of the testes. When the testes are too cold, the scrotum will retract. The muscular portion of that will retract, pull the testes closer to the body to warm them. When the testes are too warm, those muscles will relax and the scrotum will descend, allowing the testes to move further away from the body and cool. But there's also essentially a radiator that is built into the vascular supply, and it's called the pampiniform plexus. So as our testicular arteries are moving down the spermatic cord toward the testes, they are going to be wrapped by a plexus of veins. Now, as you can imagine, venous blood returning back into the abdominal cavity is going to be cooler than the arterial blood. So this pampiniform plexus, this venous blood, is going to cool the arterial blood, and in that way it's going to buffer it so that the blood entering and leaving the testes will be about the same temperature. And so this pampiniform plexus, again, think of it almost like a radiator that's going to be exchanging the heat before it gets to the testes. Here we can see the vas deferens, here we can see a testicular artery coming down, and here is our pampiniform plexus that is moving around the arterial supply. So the warmer arterial blood, the heat's going to be exchanged and moved into the venous blood as it moves out rather than make its way to the testes. All right, so a little more about seminiferous tubules. These are a number of very highly coiled tubes, and they're coiled so that you can pack them into this smaller area of the lobules within the testes themselves. As you can see, they can be upward of 70 centimeters long, so they're very long, but again, highly coiled and folded in this very defined space. Now, the epithelium is going to be the cells from the basement membrane, our regenerative cells, all the way to the lumen of these seminiferous tubules. So we can't really describe this as a stratified cuboidal or a stratified. It's its own type of seminiferous epithelium. And so we've got some collagen fibers that are also going to be present here. A lot of interstitial tissue tying all these seminiferous tubules together. So it's very different than what we see with our other epithelial types. And so we just simply refer to this primarily as our germinal or seminiferous epithelium. Now, the cells that comprise this, we're going to start with the supportive cells first, and these are the Sertoli cells. These are going to be the biggest, the tallest cells that we will see in the seminiferous tubules. They certainly are going to rest on the basement membrane. We'll see a fairly oval-shaped nucleus, and this is mostly going to be pointing in the direction of the lumen and toward the basement membrane as opposed to horizontally. Now, the interesting thing about the Sertoli cells and our developing spermatogonia and spermatids, they're all going to be joined together with occluding junctions. Now, this is critical that we have a blood testis barrier. This is going to be an immunologically isolated site because think of what's happening in the seminiferous tubules. You have meiosis. We have genetic rearrangement, genetic reorganization, and so the spermatozoa in the seminiferous tubules are going to be different than that in the male's body, and so it would be recognized as foreign. So therefore, these occluding type junctions are going to be protective of the developing spermatozoa, and the male will not mount an immune response against those cells. So here's our illustration. We can see at the bottom we have our basal lamina. Here's a fibroblast, again laying down this type 1 collagen-rich connective tissue. Then here we have this large cell. This is going to be our Sertoli cell. Now, Sertoli cells are also called nurse cells. As you can see, these late-stage spermatids, they are going to be basically implanting themselves down into pockets within these invaginations in the cytoplasm of the Sertoli cell. And the Sertoli cell is actually passing materials to the spermatids to keep them alive and support them. So hence the name nurse cells. Here we can see that we're going through different stages of development. We have a spermatogonia. This is our regenerative cell. This is going to give rise to primary spermatocytes, which will be very easy to recognize because of this condensed chromatin that we see in their nucleus. This then gives rise to our secondary spermatocyte, which we will not see. This is such a short-lived stage because this is going to quickly proceed through development from a secondary spermatocyte into an early spermatid. So if you see round cells higher up, certainly away from the base, and they do not have this very two-tone condensed chromatin, then we're going to call this an early spermatid. And what we see here with a flagellum, this is a late spermatid. I don't know why they have late spermiogenesis, but this is a late spermatid. And those are the cells that we're going to recognize in our sections of a seminiferous tubule. So here we have our basal cells. Here we have a seminiferous tubule. Here's a section through another. Down along the base, these are our spermatogonia. And there's an A and a B spermatogonia. We're not going to distinguish those ourselves. And then here we can see that oval-shaped nucleus of our Sertoli cell, the nurse cell. Here we can see a Sertoli nucleus, a Sertoli nucleus. So those are fairly easy to recognize. Here's our very condensed chromosome of our primary spermatocyte. And as you notice, we have other nuclei present here, but the majority of these are going to be our early spermatids. We're not going to see, as I mentioned before, we're not going to see our secondary spermatocytes. These are spermatids. So this is early spermatid, and these very angular torpedo-shaped nuclei, these are going to be our late spermatids. And those are the cells that principally we're going to identify within the seminiferous tubules. Now, the Sertoli cells, they're going to support the spermatozoa. They're also going to be fairly phagocytic. So as we go through our development of spermatozoa, we're going to see that much of the cytoplasm is going to be jettisoned. All the cytoplasm you are going to get during your development, you get from mom. What you get from dad is basically half a set of chromosomes. That's pretty much it. The Sertoli cells, as we mentioned, the occluding junctions help create the blood-testes barrier. But the Sertoli cells are going to also produce anti-Müllerian hormone. Now, this is critical during development. As we begin developmental processes, and this is a small overview from developmental biology next semester, but the default pathway of gender development is femaleness. Until a switch is thrown from the Y chromosome in males that begins the process of male development and materials are produced to shut down female development, such as anti-Müllerian hormone, then again the default will be female. Males have to turn on additional structures and turn off the existing female structures before male development can actually continue. Now, part of the way nurse cells support the spermatozoa is this fructose-rich medium, basically the food that's going to be provided to the spermatozoa as they continue that process of development. And now we're going to look at the process of spermatogenesis. Now, spermatogenesis represents this entire process of starting with spermatogonia and moving through the developmental stages all the way down to a mature sperm, a individual free-swimming mature sperm. Now, before the sperm leave the male reproductive tract, they are incapable of fertilization. That's going to be a last process that happens once inside the female reproductive tract. So we still have that additional step to go. So spermatogenesis is from the very beginning, our regenerative cell, all the way until we get an individual free-swimming mature spermatozoa. Now, we can break this down into a couple of different steps. First, we have spermatocytogenesis. That cytogenesis part is the key because here we're changing a spermatogonia into a primary spermatocyte. So this is the first step of spermatogenesis. Now, once we get a primary spermatocyte, then we're going to allow meiosis to convert this into a spermatid. Now, this illustration shows these late-stage spermatids, but remember, our early spermatids are going to look very similar to what we see here with our secondary spermatocytes. So I would rather see this meiosis term apply to those early spermatids, because the last stage is spermiogenesis, where we take those early spermatids and we convert them into a spermatozoa. Spermatozoa is going to be this individualized cell, this free-swimming cell. You can see late spermatids, even though they have basically this shape, they are still going to be attached via these cytoplasmic bridges. These are not going to break during this process of meiosis, and you can see how they're all connected together with these cytoplasmic bridges. And the last part of spermiogenesis is these spermatids are going to jettison that cytoplasm, disconnect the bridges, and then you'll have your free individual, free-floating spermatozoa. So let's look at these different cells and go through these steps. Spermatozoa, these are going to be our regenerative cells in the basal compartment, and they basically are going to move through a couple of different stages. We have these type A's, and these are going to be our reserve cells, the proliferative cells. They are going to move through a dark type to a pale type. This is the next step. Now, we're not going to distinguish these differences, but I want you to understand the process. Then these will develop into a type B, have a more rounded nucleus, and these are going to be the ones that immediately precede a primary spermatocyte. Now, as these cells mature and move through these stages, they're going to move up in the compartment, move from the basal surface up toward the luminal compartment. We call it the adluminal compartment. And so primary spermatocytes are going to be higher in the column. These are going to be the largest cells in the seminiferous tubules, largest of our spermatogonia spermatocyte lineages. I do not believe that they're larger than the Sertoli cells, so I think that's a distinguishing feature that we need to understand. Now, the nucleus is the most distinguishing characteristic. It's going to be very vesicular because of the condensed chorded heterochromatin, and that's really what we're going to be looking for to distinguish that. The primary spermatocytes are undergoing meiosis 1. So here in our column, again, we can see our spermatogonia along the base, our oval Sertoli nuclei, and very easily we can see this chorded, very highly condensed chromatin present in our primary spermatocytes. And remember, secondaries we're not going to see. They're so short-lived that the secondaries are just going to be a blip on the radar. But they're secondary because they're undergoing meiosis 2, that process. Now, as we leave our secondary spermatocytes, here's where we move into spermiogenesis, converting that rounded cell into that torpedo-shaped driven flagellar cell that we know as the spermatozoa. So we're going to go through 3 phases. There used to be 4, and if you look in the older lecture guides, you'll see an extra stage. But we're going to stick with the 3 phases that we have now. And the first phase is going to be the Golgi phase. Now, in this particular phase, what's happening is hydrolytic enzymes are being processed. They're being produced, and they're being packaged into these granules. And these granules, of course, are going to be produced from the Golgi. That's what these enzymes are. They're proteins. And so we're going to have an abundance of Golgi, abundance of these vesicles present. And these are going to begin the process of forming and aggregating above the nucleus. And they're going to form what is referred to as the acrosomal vesicle. That's going to be in the next stage, or the cap. And so as these vesicles begin to coalesce into larger and larger areas, again, in this anterior pole above the nucleus, we're going to also see that centrioles are going to begin to form. These are our microtubular organizing centers. They're going to organize at the other end of the cell, and we're going to refer to this as the flagellar axoneme because these are going to be the microtubules. It's going to look very much like a cilium. As we begin to see these changes taking place, that's going to represent the Golgi phase of development as we begin to change this rounded cell into one that looks more tapered in nature. So here we have an early spermatid, And we can see we've got a lot of mitochondria. I wish they would put the Golgi in here, but this particular artist didn't. We begin to see this acrosomal vesicle, this acrosomal granule, if you will, it beginning to form, and it's going to migrate towards this compartment of the cell. So now this is going to be the head of the sperm. They are drawing this upside down. I wish they wouldn't do that. So this is what we're calling anterior. And at the opposite end, we're going to have our centrioles that are going to begin the process of forming our flagellum filled with our microtubules. And you can see we're beginning to elongate and extend cytoplasm. And again, most of this cytoplasm we are going to jettison as we get further in development. Now, from the Golgi phase, we are going to further modify this acrosomal vesicle into the mature acrosomal cap. So the next phase of development is going to be the acrosomal phase. And here we are going to form this vesicle into this very pronounced structure that's called the acrosomal cap. It contains the hydrolytic enzymes that are going to be critical for penetrating through the corona radiata and the zona pellucida that will then allow the plasma membrane of the sperm to fuse with the plasma membrane of the oocyte, and fertilization then occur. Now, it's also during this phase that we're going to get further condensation of the nuclear material. We're going to start to flatten and assume that torpedo head shape to the nucleus of the spermatozoa. And then we're also going to see that on the end where we have the axoneme, we're going to begin to migrate mitochondria down toward and around that portion of the axoneme and the beginning of the flagellum, and these mitochondria then are going to form a mitochondrial sheath, and they're going to be the engine that's going to drive the movement of the flagellum. Here we can see in this early maturation stage, or here in the acrosomal stage, you can see how the mitochondria have moved from being completely randomly dispersed around the cell. Now we're moving down toward the base of the flagellum where it attaches, and we can see in this later stage, the maturation phase, this is what we're going to organize, this mitochondrial sheath that's present. Now, the third phase is the maturation phase. This is where we finalize everything. The cytoplasm is going to get shed, which is going to disconnect those bridges interconnecting the spermatozoa together. They were tied together because of the daughter cells through meiosis, and they formed a syncytium, but that's going to be released. And when that happens, now the spermatozoa are going to effectively be immobile, and they're still incapable of fertilization, even though they are still present with the Sertoli cells and the nurse cells until they're released in the ejaculate. Now, as we've mentioned a couple of times before, the final maturation stage that allows for fertilization to actually be able to occur and makes the sperm capable of doing so is referred to as capacitation. Now, we're not going to cover capacitation. We do that in developmental biology. It's quite an extensive process, actually, and the timing of which has to be perfect to allow fertilization to happen in the ampulla. But that's the last stage, and that's going to happen again in the female reproductive tract. So once again, as we look at a section through seminiferous tubule, just to review this, spermatogonia at the bottom. Now, what would these be? We'll go ahead and give you a little quiz here. We have that condensed vesicular-looking nucleus. Primary spermatocytes. You have one even very high up in the column here. Then when we see just regular nuclei in these round cells, what are these going to be? If you said secondary, remember that's a very rapid turnover. And so, no, these are going to be early spermatids. That's going to be the tricky part, because these cells look very different from these late spermatids. And here you can see this balloon of cytoplasm coming up from this nuclear material. This is all going to be shed right at that last maturation stage. Okay, now, what is this oval-shaped nucleus? Those are our Sertoli cells, our nurse cells. So those are the components we have in our seminiferous tubules. Now, just a little bit about the structure of the spermatozoa. When we look at these mature cells, we're going to see certainly they have the head. This is where we're going to have the condensed nucleus covered by the acrosome. And within the acrosome, these hydrolytic enzymes are going to be neuraminidase, hyaluronidase. This is going to be a critical enzyme that we have present because much of the zona pellucida is going to be hyaluronic acid. And so that's going to be important to penetrate the zona pellucida. We also have an acid phosphatase. Acrosin is a specific trypsin-like protease It's going to be important for degrading through the proteins also that are present in the zona pellucida. Now, part of the process of fertilization is going to involve the spermatozoa and a receptor on the spermatozoa binding to a molecule in the zona pellucida. It's called ZP3, zona pellucida number 3. And when this interaction happens between the sperm and ZP3, that is going to signal in a secondary messenger signaling cascade the acrosomal reaction to occur. And this is going to be a very rapid degranulation and shedding of these hydrolytic enzymes into the space that is just adjacent, which is going to be our corona radiata zona pellucida. And that's going to begin the process of fertilization as the nuclear material is going to move closer to the plasma membrane of the oocyte, fusion of the sperm and oocyte plasma membrane, and then entry of the nuclear material into the oocyte itself. That then is fertilization. Here we can see an illustration, a front view and a side view of the sperm. There you can see that very tapered head that's going to be very hydrodynamic as this is swimming through the water. You can see the mitochondrial sheath at the base, the centrioles, and then in yellow we have got the acrosome, and that's where we keep those hydrolytic enzymes. Now, as we move from the head, we certainly have this connecting piece. That's where we're going to have the mitochondrial sheath. And oftentimes we refer to this as the tail portion, the head and the tail, and the neck being that mid piece. Now here we're going to have this cylindrical material. This is where we're going to have the centrioles here in this neck or the mid piece. And that's going to connect the head to the remainder of the body. And this area of this middle piece, this is our mitochondrial sheath. 9 or so dense fibers are going to be collected around here, and this is not just the mitochondria that we're calling these 9 outer dense fibers, but very large material. And this is again, I believe, going to be microtubules in nature, and so this is going to surround the axoneme, and we'll see this illustrated in just a bit. But as we move down, we are going to have a ring-like structure that is called the annulus, and this is going to prevent the mitochondria from going any further in the tail and restrict it just to this mid-piece portion, or the very beginning of the tail itself. And that's, again, is going to be the engine that is going to move this flagellum very rapidly back and forth. Now, the main part of the tail is called the principal piece. And again, this is going to have a fibrous sheath surrounding all of our microtubular fibers that are extending down through the flagellum. And then lastly, we have the end piece. And this is where we're going to basically begin to disorganize and disassemble our microtubules into just an indistinguishable part of the tail itself. So here we can see a higher look at the head and the mid-piece of the spermatozoa. We've got the acrosome at the top. Here we have our nuclear material, and in cross-section, this is what we would see. As we have this cross-section through the mid-piece, here we can see our axoneme. Here we have our outer fibers. And then as we get into the tail itself, we have our outer fibers here. And we're going to see outer fibers with the axoneme in the middle. We're also going to see mitochondria if we section through. This is the level that we're looking at. So as we section through, we'll also see sections through mitochondria present in this outer region. So those are the cells within the seminiferous tubule. Now we're going to look at some important cells that are outside the seminiferous tubule in the interstitial spaces. And so we call them the cells of Leydig, or the interstitial cells of Leydig. We're going to find these in that connective tissue, the tunica vasculosa, where we have our vascular supply. And these cells, the function of these cells is endocrine in nature. They're going to be producing testosterone. So if you see these rounded cells, single nucleus, maybe binucleated, that's not really a great characteristic of these. But if you see these secretory type cells in the interstitium, you can bet you that we're looking at our interstitial cells of Leydig. Here we can recognize seminiferous tubule. We've got our spermatogonia, primary spermatocyte, spermatids, maybe a Sertoli cell here. And then in the interstitium between, here we have a blood vessel. But here you can see these highly vesiculated cells. These are not going to be macrophages, as you might often see with this vesiculated nature, but these are our interstitial cells of Leydig that are producing testosterone. That's how we'll be able to identify them. And then these are just fibroblasts present in the stroma. As sperm have matured, move into the lumen of the seminal blah, blah, blah. As spermatozoa mature and move into the lumen of the seminiferous tubules, they're going to make their way through the remainder of the male genital tract. And the next portion of the seminiferous tubules, as they move toward the mediastinum of the testis, they're going to get into a straight portion of these seminiferous tubules, and these are going to be termed tubuli recti. So as we see the epithelium become very short, almost cuboid in nature, and at the very end of these, we're going to see simple cuboidal epithelium. These are going to be the small, short segments that we know as tubuli recti. Now, these cuboidal cells may have short, broad microvilli, and they may have a single flagellum, and these again are going to assist in the movement of the spermatozoa through the male genital tract. In this particular illustration, we can see seminiferous tubules very easily, our germinal epithelium, and here we can see one that's becoming thinner and thinner, and you can see the lumen decreases and the cells become cuboid in nature. So as these do, cuboid, and here these are fairly tall cuboidal cells, and again simple cuboidal in nature. This is going to be that straight portion, our tubuli recti part, before we move into the next portion of our genital tract, which is still intratesticular, and this is going to be the reedy testes. Now, these are not necessarily tubules per se, but almost like sinusoids. It's a anastomotic interconnecting network that's present within the mediastinum testes. This is going to be the intermediate between the tubuli recti and our efferent ductules, and they're going to have the fairly similar epithelium, this simple cuboidal epithelium, but instead of a straight tube, it's going to be this anastomotic network. And so as you look in at the mediastinum testes and you see structures like this, simple cuboidal, but there's no rhyme or reason to it. It looks like flowing rivers and interconnecting islands. That is going to be our reedy testes. There's not going to be a huge portion of this. Certainly the tubuli recti are short. The reedy testes is not that extensive. But we're quickly then going to move into the transition between intratesticular and extratesticular, and those are going to be our efferent ductules. Now, these are going to be a fairly few number of short tubules, 10 to 20 or so in nature. And this is going to lead from the testes to the epididymis, which is external to the testes. Now, the epithelium, we're going to have some ciliated cells and non-ciliated cells, and this is going to sort of be a continual theme that we're going to see as we move through the genital tract. But in these efferent ductules, we're going to have fairly tall cuboid to short columnar cells in general. That's what the epithelium is going to look like. It's going to be simple in nature, and we will definitely see material in the lumen of these efferent ductules, just as we will in our epididymis. So our efferent ductules, here we can see fairly columnar-shaped cells, simple in nature, smaller diameter. Our epididymis is going to be much larger. And there you can see the droplets in the center, this round droplets. These are some of that shed cytoplasm of our interconnected syncytial spermatids. You can see some nuclei of spermatozoa present in here as well. So that's the indication that, number one, you're looking at efferent ductule. The other indication is you're still within the stroma, the interstitial tissue of the testes itself. So this portion of the efferent ductule is going to be intratesticular. Now, extratesticular, this is really the first part where we're going to say totally outside of the testes. And here we begin with the epididymis. The epididymis is going to be fairly long. It's 5 meters of tube, but packed into a very small space, 7 centimeters in its length of the space. And so it's going to be highly coiled, and you may have seen this in comparative anatomy. But the epididymis is going to have 3 regions: the head, the body, and the tail. The head is where we have the efferent ductules joining. The tail is going to be a straight portion, and it's going to be a short-term storage compartment for the spermatozoa before we make our way into the ductus deferens. Now, this is going to transition from our fairly short columnar type epithelium we saw in our efferent ductules. Now we're going to be in pseudostratified columnar epithelium. And one of the biggest things that we're going to see in this particular epithelium are going to be the cells that have our stereocilia. Now, like with other pseudostratified epithelia, we're going to have our regenerative cells, our basal cells, but the cells that are going to help us identify this are going to be our principal cells. And these are the ones with our stereocilia. We learned this at the very beginning of the semester, but remember, stereocilia is non-motile microvilli. These cells are reabsorbing, again, a lot of the fluid that's present, leaving the seminiferous tubules, a lot of the extra cytoplasm that's present. But these cells are also going to produce a inhibitor of capacitation. This is going to be glycerophosphocholine, and this is, again, going to prevent capacitation. Capacitation is almost like the countdown clock. Once you start capacitation, the spermatozoa are only going to have a defined period of time to function before they are then rendered immobile and unable to fertilize. So you want to delay that as long as possible until you get into the female reproductive tract. So here we can see pseudostratified nature, the tall columnar cells. It's difficult to see the stereocilia on the surface, but when you see the pseudostratified nature, when you see the lumen filled with spermatozoa, you can pretty much bet money that you're looking at portions of the epididymis. Now, this is going to differ from what we're going to see with the remainder of the genital tract. As we move into the vas deferens, it's a highly muscular tube. You see some smooth muscle here, and as the previous slide noted, you do have some peristaltic type contractions, but that's principally going to happen in the next tube, the next portion of the male genital tract, and that's the vas deferens. Now again, we're going to see the same epithelium, stereociliated pseudostratified epithelium, but the difference here is the luminal diameter and then the extensive muscularis. I mean, this is a very thick wall muscular tube. Inner and outer longitudinal layers with a middle circular layer. And as we look at the vas deferens, as it goes into the body and it approaches the prostate gland, where we're going to join on with the seminal vesicle, the ejaculatory duct, we're going to have an ampulla. This is going to be a dilated end of the vas deferens. But histologically speaking, vas deferens is very easy to identify. As you can see, you're going to be hard-pressed to find a small luminal diameter that has this thick layer of muscularis of smooth muscle. So it's very easy to identify, even if you cannot look directly at and identify that epithelium at the surface. Here again, we can see portions of the epithelium. Now, this doesn't look quite as pseudostratified to me as you might see in other slides. This is one that I pulled off of the internet. You can see some that looks a bit ciliated here, but again, maybe stereocilia here, stereocilia here. So some of these do not, I think, effectively represent stereocilia as well as we might have seen in other sections. Certainly, this is not one that I would ask you to identify whether this is epididymis or whether we're talking about the lining of the vas deferens. You need to see the muscle to be able to do that. Now, as we go into the body, here we have the vas deferens entering from the right and from the left. And we are going to form what is referred to as an ejaculatory duct. Now, we are going to have the vas deferens coming in, and they are going to join together in what is called seminal vesicles. And this is near this dilated portion in the ampulla. And so these seminal vesicles are then going to join the urethra via this short connecting tubule that's called the ejaculatory duct. This ejaculatory duct, the prostatic urethra, this is all, and even the seminal vesicles, this is all happening right at the same level of the prostate gland. So sometimes it's a little difficult to see these individual structures. We saw them in an illustration. We'll see another illustration coming up. But again, all of this is happening here. And for the ejaculatory duct, if you happen to see a section through this, then what you're going to see is simple columnar epithelium, no smooth muscle whatsoever, and you're probably going to see a lot of prostate surrounding it as well. So here we have the vas deferens coming in, the ampulla of the vas deferens. Here we have our seminal vesicles, and the seminal vesicles, the ampulla of the vas deferens, this is going to be our short ejaculatory duct that is coming in, the union of the two, and this is going to join our prostatic urethra. Again, all of this is surrounded by the prostate gland. The bladder would be just behind this, so we can't see the bladder in this particular image. Now, we consider the seminal vesicles to be accessory genital glands. Certainly, the prostate is one that we're very familiar with. Here in our seminal vesicles, we're going to have pseudostratified columnar epithelium. We'll have, again, the basal cells for regeneration. We'll have the columnar cells, short microvilli, possibly still with a single flagellum, again, to help move material out of the lumen of the seminal vesicles and into the ejaculatory ducts and into the urethra itself. We will have somewhat of a smooth muscle coat. Again, the peristaltic contractions are going to assist in the express of the material that are produced. And that material is going to be a fructose-rich fluid. Now, this is going to account for 70% of the semen that's produced in an ejaculate is going to come from the seminal vesicle. And this material, again, is going to nourish the spermatozoa on their way. Because remember, spermatozoa have mitochondria. That's the engine that drives them, but they don't have really any other cytoplasmic components and organelles to speak of. They have a nucleus, acrosome, and the mitochondria to drive the flagellum. So they're going to need the fructose, the carbohydrate, to produce ATP for their trip. Here we can see this columnar epithelium. You can see how compacted these are together. We can see, and in this case, we do have some spermatozoa present in the seminal vesicles. You know, they're going to be able to swim upstream as well as downstream. And so when you have these all compacted together with this columnar epithelium, this is going to be reminiscent then of our seminal vesicle. You can also see that these columnar cells are highly vesiculated, so they are highly secretory in nature. So that's what would tip you off that you're looking at seminal vesicle. Now, the prostate gland, this is the largest of our glands. We also have a bulbourethral gland, but in the prostate gland, this is where we are going to basically have these tubuloalveolar glands, and maybe 30 to 50 of each of these that are going to be present. And we're going to divide these into different layers of the prostate. We're going to have mucosal glands that are the innermost portion of these compound glands. We're going to have submucosal glands, and then we're going to have the main glands that are the outermost part of this. And these are going to be expressing quite a lot of material, as we can see in these secretions. We have lipids, we have proteolytic enzymes, we have citric acid, acid phosphatase, fibrinolysin, which is going to be, again, a protease. All of these materials are going to assist in creating a more opportune environment for the spermatozoa, help protect the sperm along its pathway. And as we look at the epithelium, sort of typical for reproductive tract is pseudostratified columnar. We saw that in the epididymis. We're going to see it here. We see it in the vas deferens. But the key thing we're looking for in the prostate, we're going to have this highly folded, much like we saw with the plicae in the oviduct, this highly folded lumen in the prostate. We're going to see glandular tissue in the parenchyma, but what we're going to be looking for is prostatic concretions. And these prostatic concretions are basically crystallized and highly condensed secretory elements, such as what we listed as far as being secreted. So here we see the prostate and its association with the bladder. It's at the inferior portion of the bladder, just at the beginning of the urethra. Here we can see a cross-section through the prostate, and we can see we've got the mucosal glands, we've got the submucosal glands, and then the main pancreatic glands on the outside. So main pancreatic glands would be much like the cortex part, whereas the others are going to be more medullary in nature. They're calling them mucosal. And then here we can see a lot of the prostatic ducts and a lot of this sort of interconnected, highly folded network. And in a section, again, we're going to be looking at, this appears almost simple cuboidal in nature, but when you see these crystalline concretions in the lumen in the center of these acini, of these tubular glands, that's going to be the key that you're looking at a prostate gland, those prostatic concretions. Now, the last of our accessory glands, the bulbourethral glands, we're not going to see these in any particular slide, but these are going to be at the base of the penis at the beginning of the membranous urethra, as the urethra is actually penetrating through the perineum of the body. These are going to have a simple cuboidal to maybe a shorter simple columnar epithelium. And what bulbourethral glands produce, this is going to be a thick, slippery fluid. This is going to help lubricate the passageway for the semen as it's expressed in the ejaculate through the remainder of the penile urethra. Now, the last portion of male genital tract is going to pass through the penis. The penis is the male copulatory organ. And so we're going to look at the erectile tissue, and this is going to be characteristic for the male penis. We're going to see that these three columns are going to consist of two columns that are called corpora cavernosa. These are going to be dorsal to the penis. They're also going to be surrounded by a tunica albuginea, this very thick connective tissue. That's going to be their boundary on the dorsal surface. Now, these may be interconnected through practically an isthmus, but you're going to have arteries and veins present here that can rapidly fill with blood. The veins can close off to trap that blood until after the ejaculation. Then the blood can drain from these erectile tissues and the penis return to a flaccid state. Now, as I mentioned, we have two corpora cavernosa, and we're going to have a singular corpora spongiosum. This is going to be inferior, also surrounded by tunica albuginea, but this is going to contain the penile urethra, and that's going to be indicative of what we see with the corpora spongiosa. Here we see an illustration of the penis being cut in cross-section. We can see the dorsal corpora cavernosa, and they're interconnected through an isthmus surrounded by that fairly thick pinkish tunica albuginea. And then we see the inferior singular corpora spongiosum with the male penile urethra present in the very center. Here we see a section through a male penis, a cross section. We can see the reddish staining, very dense tunica albuginea. This is going to be surrounding the corpora cavernosa. Here we can see a discontinuous septus, so we actually have communication. These are some of the blood vessels that are going to allow blood to quickly fill and then eventually leave this erectile tissue. Here we have the corpora spongiosa with the penile urethra present in the very center. The remainder of the penis is going to be similar to integument in nature, hypodermis, dermis, and epidermis. Now, the last thing that we have is the end of the penis, the glans penis. Often this is going to be covered by the thin skin, certainly the bulk of the penis is, but on the very end, we have this continuation of the skin that's called the prepuce, also called the foreskin. And this is going to be identical to what we see with our thin skin, stratified squamous epithelium. However, the prepuce, especially on the side of the glans penis, this is going to have stratified squamous parakeratinized epithelium. This is the tissue that at the birth of a male can undergo circumcision to be removed. Some, it's thought, is a hygiene issue. For some, it is going to be a religious issue, and others, it's going to be a cosmetic issue. But certainly, that is a choice of a parent. For some, it's a very delicate choice and a very difficult choice to make. However, I think by and large, it is personal preference and cosmetic as we look at today's society.